So, welcome to the new session of Neet Pro. We are currently continuing still the unit of life, and today we will start from inclusion bodies, and this is the third lecture of this chapter. So here we start from the inclusion bodies. Okay, inclusion bodies. So yesterday we have ended due to a technical glitch, and I'm sorry for that. So inclusion bodies, where the inclusion bodies are generally found, those are found in the cytoplasm, found in the prokaryotes. These are membraneless, so you can say it that ribosome inclusion bodies are membraneless organelle. Membraneless. What are the functions of the inclusion bodies? They generally functions to store reserve material for the plants. Store reserve materials. Okay. So example of the inclusion bodies are phosphate granules. I'm giving the examples. Phosphate granules, glycogen body, cyanophagian granules, gas vacuoles, so what does the gas vacuoles do? See, vacuoles are the empty spaces, and gas means those are filled by the gases. So gas vacuoles generally provide the buoyancy of the cell. Provides buoyancy for the cell. Okay. Gas vacuoles are generally present in the purple sulfur bacteria. Gas vacuoles are present in purple sulfur bacteria. Okay. These gas vacuoles, the presence in the purple sulfur bacteria, those are photosynthetic in nature. Okay, this has came into Neet 2020. These gas vacuole, where the gas vacuoles are present, in which bacteria do we find the gas vacuoles? This question came in Neet 2020. Okay, so keep it in mind. Small questions can come from these topics, and those all will carry 4 marks. If you make a wrong attempt, -1. If you do a right attempt, +4. One wrong attempt can make you from MBBS to BDS. Okay, now we come to the prokaryotic cell. Okay, the prokaryotic cell. The prokaryotic cell, let us draw structure first. So the outer wall is the glycocalyx here. The outer wall here is the glycocalyx. I can say it as glycocalyx. This is glycocalyx. The inner wall, I can say it as cell wall. Okay, and the blue structure, this one, is now nothing but the cell membrane. Cell membrane. Okay, so glycocalyx plus the cell wall and the cell membrane, what does it form? Cell envelope. So if the question comes as what is cell envelope, you can say that glycocalyx plus cell wall plus the cell membrane consists of cell envelope. Okay, cell envelope. Cell envelope. So what is cell envelope? Glycocalyx plus cell wall plus cell membrane. These three consists of cell envelope. Okay. So cell envelope has three layers. What are the three layers? There are three layers. Chemical composition of the cell envelope is complex. Chemical composition is complex. Most prokaryotes are present in most prokaryotes. Present in most prokaryotes. Firmly attached or tightly attached. Tightly or firmly attached. Acts as a protective unit. Okay. So next comes glycocalyx. So what do you mean by glycocalyx? Glycocalyx. So what do you mean by the glycocalyx? Glycocalyx. What is the composition of the glycocalyx? Composition of glycocalyx, polysaccharide. Okay, glycocalyx is made up of polysaccharides. Okay, if the polysaccharide is hard, bracket tough, and glycocalyx polysaccharide can be soft, loose. Hard, if it's hard, it's called capsule. If it's loose, it's slime layer. Thickness and composition. Thickness and composition. So it may vary. So glycocalyx provides the sticky character. So what does the glycocalyx provide? Glycocalyx provides the sticky character to the bacteria. Provides sticky character. Provides sticky character. It has came in the Neet. Okay, understand? This question has came in Neet. Cell wall. So what does the function of the cell wall? I have said the function of the cell wall. Now cell wall is made up of peptidoglycan. Composed of, see, I am just making you writing the most necessitated point for the examination. You don't have to study in depth, because in MBBS or in BDS you will study in depth in physiology, the cell physiology. You will study it in depth in anatomy, the human structure, that is human physiology, and these things are will study in the second year in pathology. In pathology and microbiology you will study in depth everything. Okay, so cell wall composed of peptidoglycan. Now, what does the peptidoglycan is made up of? Sugar plus amino acid. So peptidoglycan is made up of sugar and an amino acid. Peptidoglycan is composed of sugar and amino acid. Okay. So what does the cell wall function of the cell wall? The function of the cell wall is to maintain the shape of the cell. Function of cell wall. Function of cell wall is to maintain the shape of the cell. Okay. Now comes the cell membrane. Give the topic as cell membrane. So where does the cell membrane comes from? The cell membranes resides at inside the cell wall. Okay. The function of the cell membrane H2O and some solutes. Cell membrane is semi-permeable. So it allows only water and some solutes. Composed of lipid and protein. Okay. Nature selectively permeable. Okay. Structurally similar in prokaryotes and eukaryotes. Structurally similar in both prokaryotes and eukaryotes. Mesosome. Modification of cell membrane. Modification of cell membrane. Mesosome, invagination, oblique, extension of the cell membrane, extension of cell membrane. That is, now make a note as eukaryotes. I am writing eukaryotes as EUK. I have said it in earlier session also. Note, in eukaryotes, respiration happens. Respiration occurs in mitochondria. But in prokaryotes, respiration occurs in mesosome. Okay. So in mesosome, surface area increases, enzyme content it increases both the surface area as well as the enzyme content. So the function of the mesosome is to increase the surface area as well as the enzyme content increases. Okay. DNA replication and equal distribution of the daughter cell. DNA replication and equal distribution of daughter cell. See, this line is from the NCERT. This line is written in the NCERT, but it has not been explained in the NCERT. So let me explain this line. So what do you mean by the DNA replication and the equal distribution in daughter cell? Okay. Now let us draw a structure first. Here the DNA replication occurs. Let me draw it first, then I will explain. Let me draw it first, I will explain the matter. Okay. Maybe the sound is off, but I'm drawing the picture, that's why I'm not speaking anything. I will, after drawing, I will explain the matter. Okay, so here is the parent cell. This is the parent cell. Okay, this is the mesosome. This is the mesosome, and under it, in the red ink, I have drawn the DS circular DNA, that is double-stranded circular DNA. Okay, and after the DNA replication, see, 2 DNA has been formed inside the cell. 2 DNA has been formed inside the cell. Okay, then the mesosome divides and move. So after forming 2 DNA, the mesosome goes to divide and they move separate from each other. Okay, so invaginate means they draw inside the cell, and the cell membrane divides. So see, in the next step, the cell starts to divide, and in the very next step, the cell has been divided into 2 halves. 2 are the different daughter cell. Okay, and now don't ask me that why it's called daughter cell, not brother cell, not sister cell. You can call it as brother cell and sister cell, but the science calls it at daughter cell. Cell wall is made up of, cell wall synthesis is made by mesosome. Cell wall synthesis is made by mesosome. Okay, now we come to chromatophore. Give the headings as chromatophore. Chroma means color, you all know it. Chroma means color. Let us draw it, structure, and let us make it color. What should be the color? Delete. Okay, so this is the cell membrane. Okay, invagination / extension of the cell membrane is the chromatophore. So what is chromatophore? It is the extension of cell membrane. Chromatophore is the extension of the cell membrane and it is present in the cytoplasm. So present in cytoplasm. Chromatophore contains pigment. Chromatophores contains pigment. Perform photosynthesis in prokaryotes. Performs photosynthesis in prokaryotes. That is BGA. Okay, pili and fimbriae. Pili and fimbriae. Surface of bacteria non-locomotory, hair-like structure, okay. Now what's the pili? Let us draw it. Pili. Some are long and some are short. Okay. So long pili, long pili are tubular, elongated, consist of pili protein. Pili protein and are less in numbers. So the pili proteins, the long pilis are very much less in number, but the short pilis are more in number. Short, less in number. Short pilis more in number. Fimbriae present. What are the fimbriae? In morning, as we use a toothbrush, there was a bristles in the toothbrush. The fimbriae are generally look like the bristles of the toothbrush. Bristles की तरह दिखता है ये ब्रश जो दांत की सफाई करने के लिए यूज करते हो ना, इसका जो ब्रिजल्स है इसके तरह दिखता है ये जो फिम्ब्रे है. सो आई कैन राइट इट एज लुक्स लाइक ब्रिजल्स ऑफ ब्रश. Okay. Function. What is the function of the fimbriae? The function of the fimbriae is to attach the bacteria to the host or with the rock. Attach the bacteria with host or rocks. Okay. These are the functions of the fimbriae. So, if flagella is present, what does it mean? If the flagella is present, that means the bacteria or the organism is motile. That means ये चल सकता है, क्योंकि ये फ्लैजेला की तरह ये चल सकता है. जो जिसके पास फ्लैजेला है तो ये चल सकता है. Motile in nature. Motile in nature. Motile in nature. Okay. Now let me draw a thing. See, I'm drawing a structure. Okay. This and it is covered by, see, this is the cell wall. This is the cell wall. Okay. This is the cell wall. The outside is the cell wall and inside is the cell membrane. You all know it, cell membrane. Okay. So what does it? This is the basal body. This is the basal body. So what is it? The blue one. The blue one is the hook. So if blue one is the hook, and what is this then? This black one. This black one. This is the filament. Filament. So the filament is guarded by the cell wall. Filament is guarded by a cell wall. Filament is always guarded by cell wall. Filament is guarded by cell wall. This is the cell wall. Okay. Extension of cell wall. Okay. I am making you write step by step. I am not jumping any topic, so it will take some time. Okay. Have the patience, you will crack it. Flagella has three parts. Flagella has three parts. What are those? One is filament. Here's the outer one. The two is hook. Here is the middle part. And it connects between filament and basal body. And the three is basal body inside. Okay. Prokaryotic as well as eukaryotic. Flagella are different. Prokaryotic flagella is made up of flagellin protein. Flagellin protein. And the eukaryotic flagella is made up of tubulin protein. Tubulin protein. Okay. Now, out of the NCERT, I am saying that what are the gram positive and what are the gram negative. What are the gram positive and what are the gram negative? Gram positive means if we give some dye to a bacteria, dye means color. If we give put out some color to the bacteria, those bacteria who can keep the color after washing it with the alcohol, those are the gram positive. And if they loses the color and become pink, those are said as gram negative. This will be taught in the biochemistry of MBBS, and you will get to know everything of gram positive and gram negative in first year as well as in second year pathology. Okay, so if you in Neet, more than this no questions will come from these topics, and I will make you some I will give you some mind maps how to remember those topic in a gist. Let me complete the syllabus. I will make a mind map and a gist video or short video which you can revise to go through the chapter in just less than 15 minutes. Okay, for now it's up to it, and see you soon in the next lecture of this cell, the unit of life. Thank you.